Great. Okay, campers, welcome to session 11. I want to thank Mike and Barb for the invitation to come back here. This is just such a great venue. I wish this had been around a couple of decades earlier. And for all the other speakers before me and after me for framing this. I want to start the session with a quick show of hands. So how many of you in the audience would be interested in seeing 77 all-text slides in 10-point font on FDA regulations? Can you put your hands up really high? Good, because I have no interest in doing that either. So that's a relief. What I want to talk to you about instead today is strategy and tactics in making that transition through what some affectionately call the valley of death of preclinical development and getting our beautiful babies into clinical development and hopefully through it as well. So with that in mind, let me start out by saying, first disclosure, I'm not a regulatory affairs specialist. I'm a simple country immunologist who fell into neurology through multiple sclerosis and the back door. So here's my quick history and disclaimer. After my academic training in immunology, as I said, my first company was involved in creating tolerance in multiple sclerosis, and I got a drug into phase 1 through preclinical trials into MS. Biotech startups have a nasty tendency to run out of gas. So a couple of my companies have still existed. Others got a good start and then stopped. Along the way, I got pulled into business development for a while and became bipolar. I also worked with the National MS Society, helped nurture my affection and interest in venture philanthropies, worked on a consortium with Stanford and a couple of the UC schools, helping academics through translation. And through that and through a number of great collaborations and a few not so great, I have a real passion for academic discovery and translation. In spite of all this and all that I know, I co-founded a company called Thorini, which is still going, in South San Francisco. We raised about $26 million in private equity. We got a number of key funds from venture philanthropies, the MS Society, and Alzheimer's Drug Discovery Foundation, and a key U01 grant to do a lot of the talks that you've heard about. So you can get grants for translation. They really can play an important role. And after the VCs came in and raised the Jolly Rogers, I said goodbye with the grant. And currently I'm working with a group called Axosim in New Orleans. They've licensed in a brain organoid technology from Johns Hopkins. So we're going to be looking at myelination and remyelination in human cells. And also a neat trick spun out of Tulane. They are looking at nerve axons formed from iPSCs or DRGs in combination with Schwann cells. They put these into units that they've made and can look in, as you see in figure C, they can read electrophysiology across the axon. So it's not just single cell patch clamp. We can get electrophysiological data that we think is very helpful getting human tox studies and also looking for new targets in pain and neuropathy. So that's my checkered past. That's by way of saying I have a perspective, which is another way of saying bias, in preclinical development. And what I'd like to start with is a little philosophy. Why do drugs fail? We put all our energies into drug discovery and launch these or give them to foster homes, and then they fail. And they fail at different rates. And you may notice that neurology has a track record that is about average in its failure rate from phase 1. If you really don't like failure, I'd suggest you go into ophthalmology or hematology or commercial real estate. If you really like failure, Alzheimer's disease is the place for you, because the failure rate there is roughly 99.6% approximately. And of the drugs that have succeeded, arguably some of them don't work particularly well. There's a great need here. When venture philanthropy looks at this, they think about sepsis and pain and other difficult areas. But don't give up hope if you're working in AD. There's been a lot of new discoveries that I think have a chance to change our percentage rate. We may even get up to 94% failure. But again, don't give up. You may have seen versions of this slide before. Where do failures occur? A lot of it historically is in tox. Some of it's in efficacy, not surprisingly. There are other problems. Let me get rid of our magic dot here. Animal toxicity, adverse effects. A lot of this comes down to PK and tox. Those sorts of failures change as you advance through the clinical stages. But what's always concerned me about these sorts of pie graphs is they're a missing slice. And so what I would argue is that the missing slice is people. The reason drugs fail, not always, but part of the reason why drugs fail is people. And on the left, one of the reasons I think relates to what we heard yesterday about animal models. We do a lot of our studies in transgenic mice. We do a lot of our tox in transgenic mice. And then we do our trials in a human population. Well, first of all, we're not big rats without tails, not in most cases. And we don't do our trials in 20 Justin Biebers. So it's a diverse population out there, and that's one of the complicating factors. But the one I want to talk about is the one on the right, which is this is a team sport. It takes lots of different people doing lots of different things. And one of the challenges I've seen in my biotech experience is this is a relay race with a very slippery baton. And so what I put to you is the more we understand the different parts of this elephant, even if we just specialize in the tusk, the better the chances are, I think, that we'll have success. So I think the importance of sessions like we have today and yesterday and tomorrow is giving us all an overview of the parts of the elephant. And that's part of what I'd like to do today, is incorporate some of what you've already heard and what you will hear to talk about this whole elephant and what we can do to improve the rates of success. So I'll bring this up again, this slide that we have burned into our retinas of attrition in drug discovery, to make a couple of different points. And one is one that Adeline already made, which is the drug discovery process is a continuum, but there are a couple of markers along the way. The one at the end is the NDA, but what we're focused on here is getting to the IND, the investigational new drug application. To get there, we can ask the question, when does an IND preparation start? And what I put to you is, it starts really early in drug discovery as you think about what you need to do to get into an IND and plan your studies. And in fact, I think it makes sense to begin with the end in mind and think about that clinical trial and ultimately what that label is going to look like. So that's that process of winnowing down from hits to leads to drug candidate that you've already heard about is informed in part by what you think you're going to do with the IND. And the focus of today's sermon is mostly in that preclinical section. Now, I use the term preclinical. You also heard, I think, the term non-clinical, because non-clinical work doesn't stop at the IND. And I'll talk a little bit about what's going on in tox after you get into clinical trial. That continues. So again, preclinical development is a continuum. Non-clinical studies don't end with the IND. And finally, the most important reagent in drug development is time. I've been involved in programs that went through preclinical development solely based on non-dilutive funding from the government and state agencies, developed a great second-generation serum for breast cancer, and used up almost all its patent life to do so. We couldn't get a single VC or pharma company interested because by the time they got it through clinical trials, it would be completely off patent. So it's important to be efficient here. That said, nobody's perfect. We all make mistakes. The goal is to learn from those mistakes. So with that in mind, what you'll see during my talk and a number of other talks here is just a sea of three-letter acronyms. I'll try to define them as I go. You already heard a lot of them. But as we take this river cruise through the river Styx between research and clinical development, you're going to encounter a lot of these, including the IND. And I've never understood why we call it IND instead of INDA. It's an IND, investigational new drug application. We seem to have dropped the A. All I can figure is we just love three-letter acronyms. But the acronym I want to talk about is a four-letter acronym. It's one that we use a lot in preclinical development. Actually, it's not that one, though we do use it a lot. It's this other one that I want to show you. If my slides advance. There we go. That's my four-letter acronym that I want to focus on, risk. And risk drives a lot of what we do. It drives a lot of the way this is looked at by people on different parts of the elephant. I'll show you what I mean. A number of years ago, due to my bipolar condition, I was at the J.P. Morgan conference in San Francisco, the big investment conference, where a lot of the VCs and pharma come to look at new drug candidates. And a talk was given by Ed Saltzman that really struck me. We're used to thinking about proof of concept, but Ed talked about proof of relevance, and it's a way of thinking about risk, and it really stuck with me, and I just want to leave you with this thought briefly, that we're balancing all the way through this preclinical and clinical development, the balance of commercial risk and scientific risk. And a lot of what we are trying to do, a lot of what our potential partners are looking for, is how we've de-risked these candidates. And de-risking means, for one thing, we're very aware of differentiating ourselves from our competitors, especially the ones that have failed. And by the way, when you're writing a grant for translational development, it's not a bad idea to differentiate yourself from what's gone before. Why are you different? Why do you have potential advantages? We have to think about patient populations early on, alternatives, and hopefully we're learning from previous failures. But I'm going to focus here more on the scientific risk reduction, because we are, after all, scientists, and worry about how our preclinical models, especially our animal models, are predictive and how they translate into development. I've done a lot of work with EAE, the model of MS, and the challenge there is a lot of things that work in humans work in that animal model, and so do a lot of things that don't work in humans. So, and yet, when we sit in study sections, we all want to see the in vivo data. So it's a real conundrum. I'll focus mostly on this talk about safety and manufacturing scalability. But developing biomarkers can really help de-risk. It can really help your clinical trial. And don't be surprised as you're working through this, the decision of what clinical indication to go into first may change. And there are a lot of factors that affect that. Okay, so as we think about the world's toughest version of chutes and ladders, that is preclinical development, before we talk about playing the game, we're going to have to talk a little bit about the rules of the game. And so here, I promised you we wouldn't do 77 text slides, but there are a couple inevitabilities, and at least the font is a little bit bigger. What is an IND? When is it required? Well, it's actually required if you're going to take an unapproved drug across state lines. Though don't try to do it all in Delaware. That doesn't count either. An IND is required when you conduct a clinical investigation, and that's defined by the Code of Federal Regulations, CFR. There's another three-letter acronym, and specifically 21 CFR 312.3, rolls right off the tongue. It's required when you're going to do a clinical investigation of an unapproved drug, an approved drug for a new indication, or an approved drug in a new patient population. A drug is defined by the Food, Drug, and Cosmetic Act as articles intended for diagnostic, cure, mitigation, treatment, or prevention of disease. In other words, there's very specific definitions of drug and very specific reasons why you have to get FDA approval to do a trial. Those regulations, those rules of the game, are in this Bible, the CFR that I referred to. And there is a food pyramid to this. Congress has passed laws like the Food, Drug, and Cosmetic Act. Within those laws are regulations. The FDA is responsible for CFR. They're enforceable as law. The FDA also puts out guidances. They are not laws. You can ignore FDA guidances. They are strong suggestions. Very, very strong suggestions. They're good advice. Circumventing the FDA is not a good idea. If there's a reason why you want to avoid the guidance, there may be a good reason, but don't try to avoid the regulations. You won't pass go, you won't collect $200. So again, the rules for this are in Part 3112 of the IND. Non-exempt requirements. Why do you have to submit an IND? If you do a change in route, that increases risk. If you do a change in dosing, that increases risk. If you do a change in patient population, that increases risk. Can everybody see the red commonality in this? Risk. It just keeps coming up. The FDA is trying to reduce risk to patient populations. There has to be a rationale to going into this, but again, there also has to be a reason that you're not putting a patient population in risk. Be really, really nice if you showed efficacy too. Turns out that your pharma partners and your funders are really hung up on efficacy. But in the beginning, we want to do no harm. So if you're in doubt about this, ask your regulatory consultant or the FDA. There are a few cases where you don't need an IND. If there are minor variations in dosing form, or if you're using an endogenous compound, or the one that does come up from time to time is if a healthcare provider is prescribing a marketed drug to treat a patient for an unlabeled indication, the so-called off-label use. So your doctor could decide that an approved drug for A, he wants to try it in B, but he's only going to do it on that patient. If your biotech company decides that they want to tell their sales staff that they need to push off-label sales with the medical community or they're fired, that is not legal. That gets you in trouble with the FDA and the Security and Exchange Commission, and don't ask me why I know this. So, FDA divisions. For most of the drugs or candidates you're interested in, you're going to be going to CDER. Now, they're all using the same Bible, but there are some subtle differences. And the FDA has been historically divided and redivided into what clinical entity goes where. So for small molecules and for monoclonal antibodies, you go for the Center of Drug Evaluation and Research. Again, for biologics and monoclonal antibodies, you go to CDER, not to CBER, the Center for Biologics Evaluation and Research. Confusing, but CBER is responsible for blood products, vaccines, tissues, and cell and gene therapies. So know where you're going, know who you're going to, and each of these have subdivisions. But again, they're using the same Bible, and as Adeline mentioned, that Bible now is the ICH, pronounced IC, actually pronounced ICH. It stands for International Council of Harmonization of Technical Requirements for Pharmaceuticals for Human Use. And I tried to do that in one breath, and I failed. You know it's international because the harmonization is with an S, not a Z. This was developed several years ago to try to bring conformity to the regulations from the United States, Europe, and Japan, so we're all using the same Bible. And most recently, there's now a new provision so that you can get one IND that can go to multiple EU countries. You don't have to submit one to each one. So that's in transition. But this food pyramid will show you the sorts of modules that Adeline and others will refer to that are key components of this IND application. And again, the two that I'm going to really focus on today are the ones involved in manufacturing and in safety. And the way I like to think about it is the three-letter acronym SAE, Serious Adverse Event. And I like to convert that into a four-letter acronym and say that what we really need to do in getting into an IND is to show that our candidate, our clinical candidate, is sane, same, and safe. So let me really focus on same and safe today. I'll give a little bit of a mention to sane, and the clinical talk later on should come back to this. So clinical colleagues, forgive me, this is really important that you need to show some rationale with your preclinical data for why you think this is a reason to target in people, and you need to show a good clinical plan. So I'm going to take a risk here and show you a Gantt chart. The reason I'm doing this is to try to make a couple of points, but with this caveat, this is a lie. This is generic. There is no one Gantt chart that will show you how your preclinical development program is going to progress. One size does not fit all. Your results may vary. But what I want to make a point here, actually a couple, is that not only is this a team sport, but it's often going on in parallel. You don't have to do that, but you'll run out of time if you really wait and do all your manufacturing and then send it to tox. Also, the point is there are points along here where there are dependencies. You can't get to step B until you've completed step A. So there's a need for coordination here, and that coordination is critical. And also, again, as I said, I think it really helps if, it's highly unlikely that you're going to be the one doing all of this. But I do think it's beneficial if you have at least a working knowledge and hopefully a little bit of respect for the other people who are involved in this. A quick note on the other people involved in this. You don't have to do it alone. You shouldn't do it alone. And more, we're forming virtual companies or semi-virtual companies where we may not have all the components within one roof. We may be relying on contract research labs, CROs. There we go with the 3 letters again. And we also rely a lot on good project managers. CROs. There are a lot of groups from Charles River to little mom and pop shops. The problem with CROs is that there's no Yelp for CROs. So it really helps to ask around to other people who have developed and used CROs. The other problem with CROs is a lot of them are really booked. Your ability to get something done in a CRO depends largely on their timeline. Also, consultants can help you manage CROs, give you advice, put your plan together. I've done some consulting. Other people here, I'm sure, have done some consulting. One of my favorite websites, despair.com, has a nice poster about consulting that says if you're not part of the solution, there's good money to be made prolonging the problem. So, there are good consultants and bad consultants, but a good consultant can be your Virgil and your guide through hell. And the other thing I'll say about project management is a bad project manager can get you to the train wreck on time. A good project manager practices what I call preventive worrying and can see and anticipate and hopefully solve the problems before they happen. That said, there are going to be glitches along the road. So to give you some examples about this, unfortunately, I really can't go into specific companies and their pathway through preclinical development. I could, but I want to protect the innocent, and I also don't want to be sued by the guilty. So what I thought I'd do instead is talk to you about a few hypothetical examples from a hypothetical company that I like to call Sisyphus Pharmaceuticals. And those of you who are familiar with Greek mythology will understand why I gave it that name. I think it is a good example of the biotech experience. Those of you who don't know Greek mythology, look it up later. But I want to talk about their 2 molecules. Their lead molecule, FKR666, which I am not showing in its structure because it's proprietary, so we'll represent it as an ant. And their other monoclonal antibody lead, mabimabimab, which I will show you here. Please don't reverse engineer this. It's proprietary. So let's start with 666. As you heard, when we're doing pharmacology and when we're going from hit to lead candidate, we're winnowing. We're taking a lot of candidates and we're trying to juggle a lot of properties, and we're trying to look at their cytotoxicity, protein binding, CYP inhibition, induction, stability, and we're trying to come up with the best possible candidate to establish a safe starting dose, a regimen that produces the least toxicity. We're going to try to get what we need, where we need it, in an efficacious dose. So we're balancing potency with delivery, with area under the curve, and all the things you heard from Keenan yesterday that are really, really important. We're trying to get, if possible, some insights into tox biomarkers and possibly other clinical biomarkers. I borrowed this slide from one of the best translational academic scientists I know, Marty Watterson at University of Northwestern University. It's an iterative process in this early stage. We're looking at microsome stability. With a lot of our neurological candidates, we got to worry about getting past the blood-brain barrier. So we're doing things like looking at whether they're PGP substrates. We're doing KCO or MDR, MDCK to see how it goes across that blood-brain barrier, because we don't want it to be kicked out. We want it to get through. We're converting that to some in vivo studies. And in the end, we're developing something of a scorecard as we understand the various conditions, and we play something like whack-a-mole as we optimize one attribute and watch that affect another attribute. This is borrowed from the Alzheimer's Drug Discovery Foundation and somewhat modified, just to show you some of the things that we're all trying to keep track of as we develop our preclinical small molecule candidate. But as I said, we want to keep the end in mind. So one of the ways people will do this, and it's not mandatory, but it's a useful tool in some cases, is to develop what's called a target product profile, a TPP. Here we are with three-letter acronyms again. In this case, the philosophy is we're starting with the FDA label in mind. What's our final label of an approved drug going to look like? What are the clinical indications? What are the dosing? What are our aspirations? And how far are we from this? It can be somewhat flexible, but if it's too flexible, it's meaningless. So as I like to say, there's one way to hit a bullseye that's guaranteed. You shoot the arrow and then you paint concentric circles around it. If that's the way you're doing your TPP, it's going to work, but it's not going to be a good guide as you get through preclinical development. Instead, and there's no prescribed way to do this, they give a few examples in the guidance for industry that the FDA has put out. But what you do want to do is think about some of the attributes you're aiming for that will make sure that you are safe, sane, and sane. And some of that can often parallel the sections that are in your FDA 1571 table of contents. So, moving into these two areas of sane and safe, since we've done a lot in talks, I want to start with CMC, chemistry, manufacturing, and control. And the idea here is consistency, to make sure that your drug product or your drug substance can be made to the right level and the same way every time within specifications. So I like this slide because it illustrates the point. One of these things is not like the other. And if you're thinking it's the one on the left because he's taller, well, you've got a future in biotech management. But the idea here is to be able to make this little prairie dog again and again and again. So this involves transitioning. When we start with our small molecule, we may be maximizing through SAR its potency, but we're really going to be picking a candidate or two to focus on. We're going to characterize the salt forms, define any polymorphs, and we're really going to be really strict about purity, because purity has consequences. If your impurities are above a certain point, you have to report that, and you have to check them for toxicity. So the purer, the better. You're not going to exceed 100%. If you do, the FDA has got a whole other set of questions for you, but as close as you can get to 99.9 as possible. Stability is important because you need to make sure that it lasts longer than just the manufacturing plant to the trip to the clinical site. And in fact, stability will really govern how long you can use this, how long it is until you need to make more of this. And you're going from milligram amounts to kilogram. Also, the number of steps is really, really important. We might start out with a synthesis that's 10 or 12 steps, losing yield along the way. That may be okay for the bench. It's not really okay when you're getting into the clinic. So you want to minimize the number of steps for maximum yield. And you're going to end up with a certificate of assurance that I'll come back to in a moment. Oh, here's my moment. So, as you heard, we're going to be working with drug substance, and sometimes to get into clinical trial, all you're going to do is take that powder, put it in a capsule, and shove it in somebody's mouth gently. But the other thing you're going to be working on is formulation, and we just heard a really good talk about that. That you're going to be parallel working on how you're going to improve the formulation, perhaps for oral sustainability. The caveat here is formulation will never put lipstick on a pig. You're not going to improve some bad characteristics with a good formulation. But you can really improve your drug product sometimes with good help from your formulation department. So I think the talk we heard this morning was really, really important. As you work on this, you want to make sure you have analytical methods developed for two different reasons. One is for lot release. When you make your drug in your manufacturing plant, you need to make sure you're doing it consistently And you need to get what's called a certificate of analysis that shows what the parameters of this drug are, stability and a variety of other things. So your assays need to be validated. Validated has a very specific meaning here in terms of consistency, user-to-user, reproducibility, limits of detection, a lot of things that fit into a slide with 10-point font. Just to make the point that validated methods are important, and they need to be validated not just for something in an aqueous solution, but for what you're going to be doing in a biological solution. Because when you run your tox, when you run your PK, and you want to see how much of your material is still there in a messy solution like blood or CSF, you need to make sure you've got an assay that can read it in that gook. And then finally, you're going to be making GMP pilot batches and GMP batches, ultimately good manufacturing procedure. They are the manufacturing version of GLP. So, and again, there's an art as well as a science to all of this. Because as you set your C of A, you don't want your parameters so broad that they're meaningless and that the FDA comes back and says, you've got no assurance that you can make this consistently. You also don't want to make this so narrow that 4 out of 5 of your lots fail. So this has a lot of back and forth discussion and a lot of that first 4-letter acronym that I showed you before we converted to risk. Let's talk about pharmacology and tox really quickly, how we get to safe, and how we walk that fine line between safety, efficacy, and toxicity. So a couple points about our small molecule. As you heard from Adeline, one of the things that's different from the large molecule is an earlier emphasis on genetic tox. But you have to line this up with your manufacturing so you have at least GMP-like drug that you're taking into your tox studies. You're going to do some early dose range finding, PK, to help inform how you're going to do your later studies. And you're going to make some decisions. First of all, it's essential that your tox design matches your clinical design, because that's going to be what sets the parameters for what you can go into in your first phase 1 trial. Where this becomes a bit of a discussion sometimes is whether you want to take the risk and the cost of doing a longer tox study. I've talked to some of my clinical colleagues about this. They say in some cases in pharma or a big biotech, you might want to run a study out more than 28 days because you're going to be using, in most of our cases, a chronic application. And you may not want to do later on in your non-clinical studies a bridging study of a longer tox study. But in most cases, you're going to do a fairly short tox study that gets you into your phase 1. And as Adeline mentioned, the non-clinical goes on beyond the IND. In small print here, so you can't read it, shows you a couple of the other things that you'll be doing to look at more chronic tox studies to stay ahead of those phase 2 and phase 3 trials that have more patients and more time involved. And if you're lucky, you don't see this guy. But Sisyphus was not lucky. Sisyphus had 4 rats that died in the middle of a chronic tox study in the middle of the night, too late for, or unfortunately, whether it was too late or just not on the CRO schedule, those rats were not harvested in time to find out why they died. They submitted that to the FDA. The FDA said, that's really interesting. Go back and do that study and find out why those rats died. And Sisyphus decided, okay, well, we're going to do a cardiotox study to show you we think that's what the problem was. Then eventually they had to redo that whole study to see why those 4 rats died. So the clinical hold can stop you before you can get into the clinic. When you submit the IND, you have about 30 days, I believe, for the FDA to respond to your IND plan. And by the way, although I don't have a slide on it, it's often a good idea to have a preclinical IND meeting to show the FDA your tox plan and other elements you might have questions about and get some feedback. It's not required, but it can be a good idea. So clinical hold can be lots of different reasons. Lack of qualifications of your clinical investigator, misleading brochure, impurities, as I mentioned, the tox problem, clinical trial design that doesn't line up, But you really want to avoid this if possible. And that's where working with experienced people can come in handy. So the good news is Sisyphus had another drug in development. It's a monoclonal antibody. So let's take you through the differences between small molecules and biologics. Here we're really going to have to think a lot about the production cell line because the yield and consistency of that cell line is part of your CMC and part of the reason why this can take longer. Again, you're going to need validated assays. You're going to be worrying in your production line about whether you have viruses or endotoxin contaminating that, because that's really going to give you problems if it goes into humans. Cell line stability. You may have a great cell line. The second batch it produces ends up being the last batch it produces. The yield just plummets. You've got a real problem there too. So in the beginning, you're going to really spend a lot of time optimizing those production lines. And in some cases, and this used to be more true in monoclonal antibodies than it is now, that often meant licensing in some production cell line, particularly with special vectors or promoters that would really increase your yield. And because you had to license, really increase your costs. Again, you're probably scaling up to kilograms. And again, you're looking for C of A. Upstream, a lot of your time early on is going to be spent in establishing a research cell bank and then a master cell bank, which is your source to pull out an aliquot from time to time for a working cell bank, which better be consistent. Downstream processing, you're going to need a high throughput purification system. It may be in your bench you're using protein A, but that might not scale up very well. And they're going to be using various ways of viral elimination and what they call polishing to get your product out. You're again going from drug product to drug substance. You're again validating assays, coming up with analytical methods. And you're again in tox worrying about the same sorts of PK and dose range finding, but you're also worrying about immunogenicity and cross-reactivity, as you heard earlier. There is a tendency for human products to be recognized as foreign by other humans because, again, we're not all Justin Biebers. And this was a particular challenge in my first drug candidate, which was a major histocompatibility protein. It varied from person to person. It was highly immunogenic in the wrong haplotype. But this even came up with early MS drugs where the alpha interferons had a lot of immunogenicity. And one of the things we have to look at is how much of those immunogenicity reactions are eliminating the monoclonal antibody and how much of them are not affecting their efficacy particularly. So again, you're going to be looking at the clinical trial here. You're going to be wondering whether, or asking your clinical colleagues, whether the design is single ascending dose followed by multiple ascending dose or some other design, and design your tox studies appropriately. Again, non-clinical studies will continue after your IND. Those non-clinical studies are sometimes, or in many ways, different from what you see with a small molecule, and particularly with immunogenicity that can continue on well into the phase 1, phase 2 stage of your clinical development. Okay. So again, Sisyphus hits a clinical hold, this time in its phase 1, where it turns out they're causing a lot of brain swelling, and about 33% of the patients join QAnon. So the good thing for them is they've got a third drug and a third way to go forward that I want to talk to you about briefly. And that is drug repurposing or drug repositioning. And that is using finding new applications for an established drug, or if you will, teaching an old dog new tricks. Sisyphus is going to do this with an established cancer treatment called Cancer Blast. And they're going to take it into Parkinson's disease. What a good idea. What is repurposing? Repurposing is when you're applying an active pharmaceutical ingredient, API, that is already on the market, and you're going to take it into a new indication. What it is not is if you're optimizing side activities of an established drug, an approved drug. that is actually a new chemical entity, an NCE. So nice try, but back to starting square on an IND. The basis for this is diseases that might share biological targets, and that some drug activities are pleiotropic. So what this may let you do in some cases is use established studies, safety studies and others, to avoid possibly even a phase 1 clinical trial, certainly some of the early steps in preclinical, as you can get access to some of this data. This is a lot easier to do if you own that data, but it is possible to get access to some data that other people have developed. And there's a number of well-known examples of this. Perhaps the best is the little blue pill in the upper right-hand corner, Cialis, that many of you may know was discovered during the clinical trial for a cardiac indication where some of the nurses noticed that some of the patients were a little embarrassed about some of the side effects and trying to hide it. And next thing you know, you've got a blockbuster for erectile dysfunction. So repurposing can give you some really interesting results. In some cases, repurposed drugs can be drugs that were denied in the first place. Thalidomide being a good example, where fortunately the FDA caught this, but in Europe it was being used for morning sickness in the 50s and early 60s and caused a lot of birth defects. And yet it's gotten a new life through Celgene for use in autoimmune complications of leprosy. Needless to say, you don't give this to pregnant women with leprosy. But it's also been repositioned for multiple myeloma. And with dimethyl fumarate, using an example in neurology, this is being repositioned by Biogen for MS as a Tecfidra, even though it was originally banned, though it was used as a mold inhibitor for a while. It was banned because it caused allergies. So advantages: it can be cheaper and faster to build on prior data. Some of the safety requirements can be incorporated, can be used from previous studies, but patents become an issue. Because this already is been established by another company or even by your own company, that may be or probably be impossible to get a new composition matter patent. If you're getting this from another company, it's because the patent, composition matter patent has run out, so you may only be able to get a use patent. You may also get some challenges because it's obvious. But the route you're going to use to get an NDA here is something called the 505(b)(2), where at least some of the studies were not conducted by the applicant. So this all sounds great. Couple of caveats. Sometimes you can use published literature. Sometimes you can use the drug master file that's on file with the FDA to use some of the data that you need for phase 1 or maybe in phase 2A. It can be faster, less expensive. It can improve the success rate, but it has some of the limitations that I referred to. And here's an example. Actually, we just heard about Narcan, and here the same company was converting it from an injectable to a nasal spray. What the average cost of a drug, a new NCE going to approval, if you account for all failures, is, by some accounts, $2.6 billion. The average cost of an approved repurposed drug is $300 million. There's a decimal point difference there. So it's very attractive. Also, the FDA offers 3 years exclusivity on new uses for marketed drugs, so that can help the patent estate. EU has a similar regulatory route, and there are also orphan indications that can give you extra exclusivity. And industry has a number of initiatives to try to encourage this, as does the NIH. But if you take a closer look at what's been successful, and this is a publication looking at repurposed or 505(b)(2) submissions from 2012 to 2016, the majority of those fall into 3 categories: new dosage forms, new combinations, or new formulations or manufacturer. New indications are really pretty rare. They can be done, but for reasons I mentioned, it can be tricky to treat that old dog, new tricks. And here's a case in point with poor Sisyphus. As I said, they decided they were going to take Cancer Blast and move it into Parkinson's disease because there was a target that looked like it might be in common, and you might be able to build on some of the data that Cancer Blast had developed. One obvious problem here that actually I think Adeline alluded to in her talk is there's a very different therapeutic index tolerance for cancer studies that can be often very acute and very life-threatening than there are for chronic neurological diseases, especially given to an elderly population. So that's one strike. Turns out in this case too, Cancer Blast was specifically developed for this indication for extra safety so that it would avoid passing through the blood-brain barrier. Can anybody think of a reason why that might be a problem for Parkinson's disease? Yeah. So why do drugs fail? Toxicity, efficacy, people. Talk to your clinical people. Remember what your label's going to look like, and try not to be stupid. So, in conclusion, and so that we can all get to lunch, understanding the drug discovery development process can really help improve the odds. I like this slide particularly. It's borrowed from a publication in Nature in 2018, and they're talking in this article about learning from failures, and I think that's really important. But the other point I think this makes is we all have our own piece in this. Understanding all those pieces, I think, can really help in the process. Not to mention, there's a lot of strategic thinking involved in this and lots of opportunities to take what you've learned in academia and apply it here. And so finally, in conclusion, part 2, begin with the end in mind, reduce risk, support your clinical design, learn from the mistakes of others, and finally, don't screw up. And with that, I'll drop the mic and thank you. Take any questions, and let's go have lunch. Thank you.
Thanks. In the repurposing section, you mentioned a drug master file, and I'm just curious how much of FDA information is public? I mean, information that has been submitted to the FDA in support of a compound.
How much of the drug master file is public? There are obviously elements of drug development that don't make it into that master file. I think the key elements of manufacturing and toxicity are involved in that master file. But this is one of the reasons why I think repurposing is easier if you're the company that developed the first one. So it can also be easy if you're working as a partner with the folks who developed that master file. If you're going to go it on your own completely and do that, you can, but it's tougher because, it's like, in some ways, the protocols that are developed and published in the methods section from somebody else's lab, trying to incorporate those into your own lab, it can be really tough to start, and go from the recipe. So I would say it can be done. That's the whole reason for having this. But if you can do it along with somebody who's developed that makes it a whole lot easier.
But Ted, is the master file itself public? I don't think it is.
I don't think it is. I may have been misspeaking here. But
you know where you can get a lot of information publicly is in the review documents. When the FDA reviews a new drug application, the new drug application itself is confidential, so you won't have access to that. But oftentimes, quite a lot of the information is excerpted from the new drug application in the reports that the FDA reviewers generate. And all of that is up on the FDA website for more recently approved drugs. So they're filling in with the reviews of the older drugs, but a lot of times very old drugs, there's very limited information. But for the new drugs, there's a lot of extensive information.
Thanks, Mike. If I can build on that real quick, too. There have been a number of initiatives to try to get pharma and companies to share their failures. So from a group in Arizona that the FDA has sponsored, the CPATH initiative that's looked at failures in toxicology, is, I think, more important than ever to share our failures as well as our successes. And thanks for the correction.
Throughout this drug discovery process, companies are dependent on products from other vendors like tissue culture suppliers and animal suppliers and fine chemicals, et cetera. How do you minimize the risk of those vendor products not performing up to expectations. You know, I guess I'm motivated to ask this because in my academic lab, very recently, we've had a number of surprising experiences of poor quality products that have negatively impacted our research. So I guess what I'm asking is there a Yelp for vendor products?
Is there a Yelp for vendor products? I wish. To my knowledge, no. There is word of mouth. So I know from my own basic research, there are certain antibody companies, and I'm not going to name them. I'm really tempted. And the more I talk to people, they say, "Oh yeah, them," where the monoclonal antibody that you use for research purposes is too variable. So keeping control of consistency is really important. And this is where I think a CRO or CMO becomes really important. There are certain CROs or CMOs in certain companies that might, or countries rather, that might do something cheap and fast. And you can generally get cheap and fast, but you rarely get cheap, fast, and quality. And so I think that doing your own audits is one way to ensure against that. So it's an important question. You are going to certainly involve other people in the scale up of this, although I've known of some investigators transitioning out of academia who thought they could do all their scale up within their lab and set up a CMO in the corner of their lab. It's not a good idea. So yeah, I think that running audits, talking to other people who've had that experience can really help you avoid that problem.
Yeah, just a quick comment on that point. If you're going to be formulating a product that is going to be administered to humans in the early stage of clinical development, it's going to have to be manufactured under GMP requirements, as we've talked about in the class, and that includes all of the excipients. So that means you're going to be using GMP-grade excipients, and those are going to have the same strict regulation that the API that you're producing is also going to be subject to. And as is pointed out, each one of these excipients is going to be listed at the FDA in a drug master file. You might not have access to that, but you'll be able to reference that, and the FDA will be able to go to that master file and get the information from the rigorous evaluation that it has been subjected to. Jim, you had a comment?
I wanted to add it to your comment about orphan product designation. Many of you are working on diseases that qualify as rare. In the United States, rare is if it affects fewer than 200,000 people, or if it's an episodic event, fewer than 200,000 people have at least one event per year. That's number one. Number two, you can file for your designation very early on. For example, in the case you were talking about, if your patent lawyers are saying, we're not sure, that doesn't prevent you from going ahead and filing, and you need two pieces of information. You need at least theoretical evidence that your product is likely to work for the indication, and you need to show beyond a reasonable doubt that the population is fewer than 200,000. You get 7 years of market exclusivity at the time of approval.
Right.
And it blocks the molecule in the indication. So if somebody else came up with another formulation of the same molecule and they can't demonstrate it's better in any way, they're prohibited from marketing.
Exactly, and I went light on that because I think we'll have that come up again tomorrow during the patent discussion. But very good point, thank you.
Yeah, maybe two quick comments. One with regard to repurposing. It is, if one engages with the company that holds the IND and they agree to partner, then you can also reference their IND. Instead of starting everything from scratch,
Yep.
you can use sections of their IND and simplify your own submission. And if a company discontinued a compound, they may be happy to partner because if they don't have any, like if they don't need to invest anything, but part of the partnering agreement means that if you're successful, they get future milestones, then that's an incentive for them to partner because no skin off their back. And then you can have the opportunity to reference their IND and have access to this information. That's one thing. With regard to quality of CMOs, CROs, I think that in the pre-GLP sort of talks stage, it's very difficult to ensure quality. It really is going to be word of mouth. If it's research grade material, you're just going to need to get reference from somebody that did it and had good or bad experience and make a decision. When it comes to GMP material for GLP tox studies and in clinic, then facilities, CROs, CMOs undergo audits from the agency. And the reports of these audits are available. And when there are violations, then they're documented. And so you can have access to that. This is public domain. And so for the research space, you have a problem. For the controlled space, GMP material, you can actually go in and see if a place has issues, and then you can make decisions accordingly.
That's a very good point, and you also want to make sure, if they've been written up, that you're aware of that.
I have one quick comment on the repurposing. It's been experience that I've observed that if a company has a blockbuster drug and you want to work with them to repurpose it, it's unlikely they're going to want to work with you, because any new information that could negatively impact their money makers is not necessarily going to be in their best interest. So it's just something to kind of keep in mind.
Yeah, I think that's a good caveat. And there might be some cases where you go to a big owner of a blockbuster drug and say, I've got a new indication, and they'll say, that's a great idea, we'll take it from here. And in the other cases, they might want to be careful about somebody running away with that and doing a lot of off-label use and discovering a brand new toxicity, which might tarnish the reputation of their big blockbuster. So there are commercial risk reasons why somebody might be less than thrilled with your idea of repurposing.
Okay, other questions for Ted? Not hearing any. I just see that Guy Alon has come back, and we had a question for you, Guy, in a previous talk on the toxicology topic, and I think it was raised, and I'm just going to try to paraphrase this. Whoever asked the question, if I get this wrong, correct it. It was, yeah, you had a question about whether testing of antibodies that are humanized, if you went into rodent to do the testing, whether you want to use the humanized human antibody or the rodent analog of that antibody. But maybe I got that wrong.
No, that was the question. Like, it's a different thing, but it might be more analogous. So
Yeah. Okay, so just chronologically, usually what's going to happen is you will start with likely a rodent antibody, and you will humanize, because humanization takes time, work, and money quite a bit. And so you will usually do your preclinical, PK and efficacy studies to gain confidence that you actually want to move forward and invest in humanization. I don't see how you start with a human and then go back to rodent. But if you wanted to, let's say that you had your humanized antibody and you wanted to do something in a rodent, you could do that. I would say that it will be easier to do if your study is, like, let's say that you want to confirm a PK profile in rodent before you move forward. Then if it's a single dose study, no problem. If it's repeat dose study, then with repeat dosing, you have an increasing chance of mounting a species-based immune response. Even if that happens, it doesn't necessarily mean that it's going to negatively impact your results, because usually the ADAs are benign. That being said, you will still need to monitor your PK carefully, just to make sure that if an ADA is mounted, that it doesn't result in faster clearance of your drug, which can then impact kind of any pharmacodynamic and or efficacy data down the road. So I think that the first question is, why would you want to test a human antibody in a rodent? Like, is there a good reason for it? If yes, then it can be done. You just need to be kind of, I think, careful with your PK. With tox, I think that the probability of that is low because usually ADAs do not result with adverse events. If anything, they can result with just a faster clearance of your therapeutic, which can usually easily be overcome by overdosing because then you characterize the PK profile, you see the elimination curve, and you dose above the clearance, and that's how you compensate. Because antibodies are usually safe, you can just up the dose to compensate for the faster clearance, and that is unlikely to result with an adverse event, although there's no guarantee, right? But usually it doesn't happen.
So the answer to the question is that, let me ask very specifically.
Evelyn, take the mic.
So the question, I think, was what do you take into your GLP tox studies? Is that correct? And I said what you're taking into humans.
Yeah, that is correct. Yeah, the one thing that you do want to do, if you're going to take an antibody into the clinic, you actually want part of your GLP tox package to be in a non-human primate. And then that is actually the driver for kind of safety and PK and dose projections for a phase 1 in human. So you are not going to rely only on a rodent PK profile. You are going to also test your antibody in cynos usually, and then you're not bumping into the issue of a humanized antibody in a rodent. So usually your package will include the rodent antibody in a rodent model,
Yeah.
and the humanized in a cyno.
No, I'll just add, when we did a biologic for multiple sclerosis, we used a transgenic mouse expressing that biologic to address that very problem.
Yeah, I think the issue came up also the potential differences in the target in the rodent versus the human. So if you go into a non-human primate, there's a less likelihood of that being an issue as well.
Correct.
Yeah, James.
That's really the issue, and it's not really the tox issue, it's the on-target effect. And so there's this famous Tegenero as a company, and they came into the clinic and they did tox by the books with taking their humanized antibody against a human target and went up, and then they came into the clinic way too high, and they had this cytokine storm in the healthy volunteers with the first dose because they started way too high, but that was all on-target effects. And so, actually, as was mentioned, one thing that you do is you're studying the effects of your antibody on the target. So obviously you've done animal studies, so you ought to know what the targets. And now, basically what we do is just start much lower. If it's a target that's never been engaged in people before, we don't just start at 1/10 or 1/100th of the tox dose, which is what Tegenero did. So that was a lesson to everyone about. But that's really on-target toxicity. There's really not off-target toxicity from antibodies for the most part.
Great. Okay. Thanks, James. So we have a few minutes before lunch is going to be set up. So I think what we'll do is open up to any questions, comments that people might have for any of the instructors who happen to be in the audience. Anybody? Yeah.
I wonder if somebody could clarify the off-label versus a clinical trial. So let's say we're taking a case where there's no biotech company involved, so there's no financial conflict of interest. It's just a clinician. What's the difference between them giving a patient a drug and paying attention to how it turns out and making note of it or publishing about it, as opposed to not doing that?
Right. Well, it's a very interesting issue, and it's not completely cut and dried. But the question was, if you have an approved drug, it's approved by the FDA for a specific labeled indication in a set of patients at a specific dosing regimen, are you able then to do a clinical trial either for an academic purpose or for registration purpose to get a new labeled indication? And I think the way it works is that it really depends on whether you are going to be subjecting patients that might create a greater risk than what was the situation for how the drug was approved. So if you're going into the same set of patients with the same dosing, then if you're doing that for academic purposes, generally you'll get an exemption from the FDA. Now you may have to go to your local authorities, your IRB, to get approval, and there may be other types of committees that you would have to present the research to for approval. So at UC Davis, we have an IRB requirement, but our chief medical officer also has a requirement for the use of experimental therapies beyond what they were intended in normal clinical use. So could be local requirements, but generally the FDA will exempt that. Now, if you're attempting to get approval from the FDA for a new indication, then you always have to go to the FDA and have that be done under an IND so that they're aware of what you're doing and what you have learned in your studies, that they can use that in their approval of that new indication. The other situation that can apply is if you're going into a different population. Let's say the drug's approved in adults and you want to study it in children, or you're going to use it for a completely different use. It's approved for one thing and for cancer, and then you want to do it in Parkinson's disease. It may present different risks when you're using it in the way that you're planning on studying it experimentally. In that case, generally what's done is you would go to your local IRB, but also the FDA would have to weigh in that situation if there was an extra risk that you were subjecting the subjects to. But this doesn't apply if you're simply using the drug in clinical practice to treat a patient. And the reason is that the FDA doesn't regulate the practice of medicine. That's done at the state levels. So in California, it's the state medical board that regulates the practice of medicine. So if you can gain access to the drug just by prescribing it at an ordinary pharmacy, then a practitioner in the state of California, for example, could apply it for something completely different. And that's actually done all the time, and it's not a violation of the law, or it's not outside of ordinary medical practice if you're doing it based on what you believe is appropriate scientific foundation. Does anybody have any other comments about that? James, that may come up a lot in your situation.
I'll just say for child neurology, pretty much everything's possible.
Yeah, no, so I think you put it well, and it's a total gray area because really an IND is permission to take a drug that's not been used in people into the clinic, right? That's an investigational new drug. And so when you have a drug that's freely available and being used off-label, then technically you don't need an IND. And I think the most important thing is actually ethics committee and IRB approval, because that's really where people get in trouble, is they go in their clinic and do a little clinical trial of their own, and then things go wrong, and then every eye in the world is looking at you saying, did an ethics committee approve this? No, this was unapproved human experimentation, and that's even with, anything, even, nutraceuticals, things you buy in a. So I would always be very careful about ethics committees. And then you could talk to the ethics committee but like you said, if you go to the FDA and say, yeah, I want to do this trial of an existing drug for a different disease, they'll just exempt you because you're not planning on trying to market it for that. Because that's what an NDA is, a new drug approval, is a new drug for marketing. So if it's already approved, there's no need for for marketing, only if you're a company or you're actually going to try to sell it.